This is an Edexcel GCSE Bio Talk Through Paper 1 from June 22. Gregor Mendel studied inheritance in pea plants. Pea plants can either produce yellow pea pods or green pea pods. Mendel cross plants that always produce yellow pea pods with plants that always produce green pea pods. A represents the dominant allele, lowercase a the recessive, which is the genotype of the pea pods produced from this cross. So you're going to have big A representing one of the colours, small a representing the other colour, so it's going to be C. I'm not sure which one yet because I don't have enough information to work out which one's dominant and which is recessive. Mendel grew many plants from the seeds in these pea pods. These plants are then crossed with each other. The seeds from the second cross produced 5,496 with yellow pea pods, 1,832 with green pea pods. Give this as a ratio in its simplest form. So I now know that yellow is dominant. So it would therefore be represented by the uppercase A. Green is recessive, so that would have been like this. Give this as a ratio in its simplest form. So we need one of these numbers to equal 1. So divide both numbers by the smallest number, which is 1,832. So 5,496 divided by 1,832. So there's my ratio 3:1. Complete the Punnett square to show the outcome of a cross where both parent pea plants are heterozygous. That means that they have different alleles. Show the percentage probability of homozygous recessive offspring in your answer. So the fact that it's heterozygous means we have these alleles. So here are our 4 offspring. So in terms of our percentage probability of having homozygous recessive, it's homozygous, 2 of the same, recessive, the lowercase allele. I can see that's a 1 in 4 chance, which as a percentage is 25%. Some plants reproduce sexually. Give one advantage of this type of reproduction because it creates genetic variation in the offspring. Name the process that forms gametes for sexual reproduction. So we're looking for a type of cell division. Is it mitosis or meiosis? You have to learn that to produce sex cells, gametes, it's meiosis. DNA molecules contain base pairs. Describe how the base pairs are bonded together in a DNA molecule. So we need to talk about the fact that there's complementary base pairing. And let's say what matches up with what. So it's the straight letters that go together, A to T and C to G, the curly letters. And then the bases, remember, are held together by hydrogen bonds. And I've actually said 3 separate points there. Figure 1 shows part of a DNA molecule. Write the code for the complementary DNA strand. So we are always following these rules. So every time you see an A, you write T. T, you write A. G, you write C. And that's done. 3 bases code for each amino acid, which is the maximum number of amino acids that can be coded for by this strand of DNA. So we know we need 3 bases per amino acid. So there's 1 amino acid, 2, 3, and 4. So the answer here is 4. That option there is B. What is the shape of a DNA molecule? Remember, it has a characteristic double helix shape. A student wanted to extract the DNA from fresh peas. The student crushed the peas, added washing up liquid and water. The enzyme protease was then added. Explain why protease was added. What does protease do? Well, it digests protein. In the nuclear membrane, because remember, the DNA is found within the nucleus. The mixture was then heated and filtered. Finally, the student poured the filtrate into a test tube and ice-cold ethanol was poured down the side of the test tube into the filtrate. State why ice-cold ethanol was poured into the filtrate. So that's to allow us to see the DNA. Basically, it precipitates it. Figure 2 shows the number of people diagnosed with sexually transmitted infections, STIs, in the UK during 2017. State the sexually transmitted infection that has a median number of people diagnosed. So we need to put these numbers here in order, starting from the smallest. So that's 0.1, 0.6, 0.8, 1.1, 3.7, and then identify the middle number, it's 0.8, which corresponds to gonorrhea. The population of the UK in 2017 was 66 million. Calculate the total number of people diagnosed with chlamydia in 2017. So we're looking at, it's 3.7 per 1,000 of the population. So we need to, first of all, get our 66 million, divide it by 1,000 to get 66,000, and then times it by 3.7. So our final answer here is 244,200. State why chlamydia can be described as a communicable disease, because chlamydia is caused by a bacteria and can be spread from person to person. Give one way in which the transmission of chlamydia can be prevented. I'd just write wear a condom. It creates a barrier stopping the movement of that bacterium. Explain why chlamydia can be treated with antibiotics. Remember, antibiotics kill bacteria. Chlamydia is a bacteria. HIV is another sexually transmitted infection. Explain how HIV can cause the onset of AIDS. Remember, HIV infects lymphocytes, white blood cells, and that's a huge issue because it compromises the immune system. So it means that normal infections like colds become a lot more serious. Figure 3 shows 2 light receptors in the human eye. Which part of the eye contains light receptor cells? It is the retina. The retina contains photoreceptors. These cells require energy. The cell organelles labelled X release energy during respiration. Name the organelles. They are the mitochondria. The cell organelle labelled Y contains chromosomes. What must that be? It is the nucleus. Don't let those awkward cells confuse you. Cell A responds to dim light and is responsible for night vision. Name cell A. It's the rods. Describe how the role of light receptor B is different from the role of light receptor A. So A is rods, B is cones, and cones help us see colour. The optic nerve carries information from the back of the eye to the brain. The optic nerve is 47 millimetres in length. The nerve impulses travel at 75 metres per second. Okay, we've got issues with units straight off. Calculate the time an impulse takes. So they've given us the equation. What I want to do is get 47 millimetres into metres. The way in which I do that is by dividing by 1,000. So I'm going to have 75 equals 47 over 1,000. And then I'm going to divide that by x because that's what I'm after. So just sort this out a bit. I've cross-multiplied to get x on its own. So my final answer here is a tiny number, 0.000627 to 3 sig fig. The impulse travels to the occipital lobe of the brain. The occipital lobe is labelled in figure 4. Which part of the brain contains the occipital lobe? Use the diagram to help you. It is A, the cerebral hemispheres. State the sense most likely to be affected. If the occipital lobe is damaged, it's going to be your vision. In 2017, a new strain of Klebsiella pneumoniae bacteria was discovered that was resistant to 26 different antibiotics. Terrifying. Explain how Klebsiella pneumoniae bacteria develop resistance. This is all to do with natural selection. So let's go through what happened. So some of these bacteria showed resistance to antibiotics. Why is that? Due to mutation. These bacteria survived treatment with the antibiotics. Once they can survive, they reproduced and they passed on these resistant alleles to their offspring. State how the use of antibiotics could contribute to this bacteria developing resistant to antibiotics. So what naughty behaviours do people do? Well, they don't finish the antibiotic course. That's a big no-no. It's a prokaryotic cell. What is a characteristic feature of a prokaryotic cell? Prokaryotic cells, remember, contain no membrane-bound organelles, including having no nucleus. So the answer here is B. New antibiotics are being developed to treat the disease caused by this Klebsiella pneumoniae. Describe the stages of antibiotic development that would occur after the discovery of a new antibiotic. So we're looking at all the drug development. So we start by saying the antibiotic would go through preclinical tests, e.g. using animals or cells, followed by clinical tests, which remember involve the use of, first of all, healthy volunteers, followed by patients. And remember, it's always good to use a double-blind trial, which is when neither the patient or the doctor knows who's been given the active drug. A student investigated the fat content of 2 types of milk, milk A and milk B. Before starting the investigation, the student added a drop of oil from a pipette into a test tube of water. The drop of oil rose to the surface of the water. The student then placed a drop of milk A into one test tube of water and a drop of milk B into a different test tube. The drop of milk A sank to the bottom and the drop of milk B rose to the surface. Give one reason for the drop of milk B rising to the surface. I'd be keen to go for a bit of a physics answer there, which is that milk B is less dense. You could have also said that it contains a higher fat content. 5 centimetres cubed of milk B and 1 centimetres cubed of lipase. Remember, that's an enzyme which breaks down lipids into fatty acids and glycerol. The pH of this mixture was pH 7. The test tube was placed in a water bath for 10 minutes. The pH of the mixture changed from pH 7 to pH 5. Explain what causes this change in pH. So we have lowered our pH, it's become more acidic. Why is that? Because that lipase has digested that lipids into fatty acids, and fatty acids have a low pH. This procedure was repeated with milk A. There was no change in the pH of this mixture after 10 minutes. Explain why there was no change in the pH of the mixture containing milk A. So there must be something different about milk A. Milk A doesn't contain as many lipids, and therefore fatty acids weren't produced. Hence why we didn't see a lowering of pH. The student repeated this procedure with lipase that had been boiled and left to cool. This was added to another sample of the fatty milk B. Describe why the pH didn't change. The key word here is boiled. Boiling denatures the lipase enzyme. Don't say kills. Say what denature means, meaning the active site changes shape. And the substrate lipid can no longer bind. A student was investigating mitosis in the roots of a garlic plant. Describe how the student could prepare a microscope slide to show mitosis in the growing roots of a garlic plant. So we want to say that we use the root tip and crush using a pestle and mortar. Stain the root. It's important that we stain so that we can actually see it under the microscope. Add to a slide together with a drop of water. And then our final step is to add a cover slip. Describe what is produced when a single cell divides by mitosis. So here's your single cell. It divides by mitosis to form two daughter cells, each with a diploid number of chromosomes, which means that it contains two sets of chromosomes and they're genetically identical. So two daughter cells produced, which are both diploid and genetically identical. And that's how we get our 3 marks. The student observed 89 cells on the microscope slide. Figure 6 shows the number of cells at each stage of the cell cycle. Use this equation to calculate the mitotic index for this slide. So we'll steal that equation. We need to give our answer to 3 significant figures. So what's the number of cells in mitosis? So PMAT is mitosis. And interphase is the gap between mitosis. So we need to add these up to get 45 over the total number of cells. So add that 44 times by 100. And then to 3 sig figure answer is 50.6. The mitotic index is often used in the diagnosis of cancer. State the effect of cancer on cell division. Causes cell division to be uncontrolled. Alfred Russel Wallace travelled around Malaysia during the 1800s and wrote to Charles Darwin about the animal species he studied. His main conclusions were very similar to those of Charles Darwin, and they both contributed to the current understanding of evolution. Describe the theory of evolution by natural selection. The key thing with natural selection is there needs to be a struggle for survival. Only the best adapted individuals survive and reproduce and pass on the favourable genes to their offspring. And this is a process which occurs over several generations. Wallace and Darwin did not always agree. Darwin believed that male birds have feathers that are brightly coloured to make them more attractive to female birds. Wallace thought that female birds have feathers that are less brightly coloured, so they're more likely to survive. He explained why having feathers that are less brightly coloured increases the survival rate of females. I think the main answer here is females that are less brightly coloured are better camouflaged, are more camouflaged, so less likely to be eaten by predators. Suggests why it is more important for the survival of the species that the survival rate is higher in female birds than male birds, because the female birds are responsible for producing offspring. And the other point is that one male bird can reproduce with many female birds. Figure 7 shows the limbs of 5 animals. Describe how the structure of these limbs provides scientists with evidence for evolution. Remember, this is called the pentadactyl limb. Penta meaning 5. Because they all have that pentadactyl limb, it indicates that they had a common ancestor with a similar pentadactyl limb structure. And then the structure has been adapted based on the different function it needs to have. So in whales, it needs to swim. Horses, they need to run away from predators. So all 5 limbs have a pentadactyl structure indicating a common ancestor with a pentadactyl structure. This limb has adapted to suit the needs of each animal. There is a shortage of kidneys for organ transplants. Scientists are investigating how to grow kidneys using genetically modified pig embryos. Step 1, human stem cells taken from the patient needing a transplant. 2, human cells injected into the early stages of the developing pig embryo. The pig embryo is genetically engineered so it does not grow pig kidneys. The pig grows the human kidneys rather than their own, and then the kidneys are taken from the pig. Poor pig. State why the embryo of the pig must be engineered so it does not grow pig kidneys. I think it's really so that the human kidneys form properly, because they wouldn't form properly if there were pig kidneys also growing at the same time. Explain why human stem cells are used for this process. So the human stem cells, remember, are undifferentiated, unspecialized, which means that they can develop into any type of cell, such as kidney tissue. And the other key thing is the use of the word human. It means they're less likely to be rejected. Figure 9 shows the number of organ transplants needed and the number of donors available in the USA from 1991 to 2018. Compare the number of donors available with the number of organ transplants needed. Use the information from the graph to support your answer. Gosh, look at this. The number of donors available is extremely small compared with the number of organ transplants. Notice that the number of organ transplants needed increases rapidly compared with the much lower increase in the number of donors available. So we're just really saying what we see on the graph. So there's 1 mark. We can also see that the number of donors available is always less than the number of kidneys required. And let's state a number, e.g. in 2010, 110,000 kidneys needed, but only 17,000 donors, there or thereabouts. So statewide scientists are genetically engineering animals for organ transplants. Well, we can see this from the graph, not enough donors available. Bacteria have been genetically modified to produce human insulin since 1978. Explain how bacteria can be genetically engineered to produce human insulin. Amazing. So we need to identify human insulin gene in the human DNA. Now let's get technical. Cut out insulin gene using restriction enzymes. Now we need to deal with the bacterial plasmid. Cut out bacterial plasmid using same restriction enzyme. Insert human insulin gene into bacterial plasmid, this time using a ligase enzyme to seal sticky ends. You have to learn this off by heart. Plasmid is returned to recombinant bacterial cell and placed in a fermenter where bacteria multiplies. Figure 10 shows part of a method used to produce a bacterial culture on a Petri dish. Sterilize Petri dish in agar, pass inoculating loop through flame, allow inoculating loop to cool, use loop to collect bacterial sample, use inoculating loop to spread bacteria onto agar. State why steps 1 and 2 are necessary. This is all about killing any pathogens which could contaminate our sample. Give 1 reason why step 3 is included. Well, it's important that the inoculating loop is allowed to cool in order to prevent the killing of the bacterial sample we're interested in. A student wanted to investigate how effective 3 different antiseptics were at killing bacteria. The student was provided with an inoculated Petri dish prepared using the method in figure 10, 3 different antiseptics, filter paper discs and sticky tape. Devise a plan for the student to complete the investigation, include a control and any variables. Great, I love variables. So our independent variable, what we're changing, we're using 3 different antiseptics. What's our dependent variable? What are we measuring? Measure the diameter of the zone of inhibition produced by each antiseptic on the agar and calculate the area using pi r squared. control variables. So what are we keeping the same? We want to keep the same the incubation time, e.g. 24 hours. We want the temperature to be the same, such as 20 degrees room temperature, and the size of filter disc, e.g. 1 centimetre diameter. Let's add a description of the method. So there's our Petri dish containing agar. Here's our 3 antiseptics, A, B and C. We've got our agar jelly. So when I said measure the diameter, you're measuring this diameter here in terms of the clearance zone produced by the antiseptic, because you'll expect there to be no bacteria because it's been killed. A few extra details: wash hands, seal Petri dish using tape. You don't have to write everything, but here's a selection of things I think are important in order to get your 6 marks. Viruses can cause disease. Describe how the lytic pathway is involved in the reproduction of viruses. Right, so we start by saying that the virus inserts its DNA or RNA into the host cell. The virus hijacks the host cell machinery to produce viral proteins. DNA and RNA. And then key is that the cell undergoes lysis, releasing virus particles.